Thank you very much for the introduction, Janet, and good morning, everyone. I would first like to thank the organizers for inviting me to present my work at this conference. It's a pleasure to be here. There was a slight change in the topic of my presentation. It's still with single-cell transcriptome sequencing, but now it's looking at the inference of olfactory stem cell fate trajectories. I've had a great time working on this project with my postdoc, Davide Risso, and my PhD students, Fanny Perrodo and Kelly Street. And I'd particularly like to thank the PI for this project, John Nye, for motivating all of our work in this area. He's been a fantastic collaborator. And I've also really enjoyed working with my colleagues, Elizabeth Purdom, Jean-Philippe Vert, and Nir Yosef. And the work that I will present was funded partially by the BRAIN Initiative and the California Institute for Regenerative Medicine. So I will discuss three main topics in this presentation: normalization, clustering, and the inference of cell lineages and pseudotimes. So first, a few words about the biological question. The main person behind this project is Russell Fletcher, a postdoc in the Nye lab at Berkeley. So neural stem cells are of particular interest in neurobiology because they stay active in certain areas of the brain for the entire life of an individual. And we'll focus on the mouse olfactory epithelium, or OE in short, as a site of active neurogenesis in the postnatal animal. These olfactory stem cells support the replacement of olfactory sensory neurons, as well as non-neuronal support cells like sustentacular cells. And they can reconstitute the entire epithelium following injury. The OE is a convenient system to study because of its accessibility, and also because there are a limited number of cell types. And we'll be focusing particularly on horizontal basal cells, and olfactory sensory neurons and sustentacular cells. So here's a diagram of the head of a mouse, and you have the epithelium here in blue. And this diagram zooms in on the epithelium and to show you the main cell types that you have in the OE. So you have the HBCs, horizontal basal cells, GBCs, globose basal cells, and these are olfactory receptor neurons, immature and mature. The HBCs can either self-renew or differentiate into sustentacular cells or neurons. So our collaborators' questions included the following. They were interested in determining the stage at which neuronal and non-neuronal lineages diverge, characterizing intermediate stages of cell differentiation, and unraveling the genetic networks and signaling pathways that promote self-renewal. So they used the p63 regulation of HBCs as a way to study differentiation. So HBCs, again, are horizontal basal cells, and they're adult tissue stem cells. The p63 protein promotes the self-renewal of HBCs by blocking their differentiation. So you can think of p63 as a molecular switch that decides between two different types of cell fates: self-renewal or differentiation. So Russell undertook a series of single-cell RNA-seq experiments to study cell fate trajectories in the OE, these HBCs of the OE, in p63 knockout mice. So this is a diagram summarizing his experimental design. He sequenced RNA from 636 single HBCs. 100 or so of these cells were from wild-type animals, and 534 of these cells were from knockout animals with the p63 knockout gene knocked out. For the p63 knockout, he looked at several time points following tamoxifen that was just giving better temporal resolution for the regulation of p63. Another way to look at the experimental design is with this bar plot. This is a bar for a bar per batch. Batch here refers to C1 run. And the height of the bar represents the number of cells that were sequenced in that batch. and the color of the bar corresponds to the biological condition, okay, whether it's a wild-type animal or a knockout animal at different time points following tamoxifen treatment. So you can see right away that batch effects are nested within biological effects. Okay, for example, here for the knockout at 96-hour post-tamoxifen treatment, you have 3 batches. Okay, so that's a little bit problematic, as we'll see later on from an analysis and a normalization point of view. So that's a few words about the design. We'll be making use of a set of marker genes, 94 of them, that Russell curated from the literature and from his prior microarray sequencing and in situ experiments. And we'll also make use of housekeeping genes, 700 or so of these that were also curated from previous experiments. And they're expected to be constantly expressed and highly expressed across the olfactory epithelium. So we'll be using these as negative control genes. Before showing you some data, I just want to say a few words about a topic that I feel very strongly about, and that's reproducible research. More specifically, that's computational reproducibility. So simply the ability to reproduce the quantitative or the numerical results of a paper. So given the data, the software, are we able to get the tables, the figures? And this is a much narrower, much simpler notion of reproducibility than biological reproducibility, which would refer to the ability to reproduce the results from one paper or one study in another study. So in order to ensure the computational reproducibility of our results, we're using a version control system to develop our software and to analyze our data. We're using GitHub, but there are other options. We're also storing our data in our data packages that have a standard object structure, in particular S4 classes like expression set or summarized experiment. And that allows us to keep together in one object not only the gene expression measures, but data that relate to the gene and data that relate to the samples, the metadata, if you want. And for authoring our tech reports, our internal reports, and our publication, we're using this framework for publication, which is called a compendium. And a compendium is an integrated and executable document, an input document, that contains all the pieces for a publication. So it's not just the text, but it's the data, the code, and the software. So by having these integrated and executable input document, you generate dynamic and reproducible output documents that mix the text, the code, and the code output. We're using, for this purpose, software that's related to R and LaTeX, and in particular, it's implemented in the R Studio. Okay, so let's look at data. And first, the issue of zero inflation. So the plot on the left is a bar plot of the proportion of genes with 0 count in a given cell. So we have 1 bar per cell, so 600 or so bars. And I've colored the bar by the batch, or the C1 run if you want. Okay, so you can see there's a huge proportion of genes with 0 counts. Huge zero inflation. So that's very problematic. This proportion of genes with 0 count is much higher than what we see in bulk or in a seq experiment. The 0 counts could occur for biological reason. The gene is simply not expressed, or for technical reasons, low capture efficiency of the technology. It's advisable to do some preliminary 0 count gene filtering before normalization and any type of downstream analysis. Most normalization methods involve scaling, and they'll do very poorly if you have a lot of genes with 0 counts. In particular, a widely used method implemented in the biocontroller R package DE-seq discards any genes that had zeros in at least one sample. So if we applied directly this DE-seq normalization method, we would be basing our normalization on 5 genes only out of 20,000 something genes. Okay? Other normalization methods also have problems with zeros. That's the case of full quantile normalization that's based on ranking the gene counts. So when you have many zeros, you have issues with ties. So as a first step, we applied the following 0 count gene filtering to our data. we retained only the genes that had at least 20 reads in at least 40 samples. So that cut by half, roughly, the number of genes that we're looking at. So just over 9,000 genes. And this is the same bar plot after zero-count gene filtering. Still a high proportion of genes with zero count, but a little bit more manageable. We're also keeping track of a variety of sample-level quality control measures. So those are the types of measures that refer to the alignment and the sequence composition of the reads. They're produced by software like Picard tools. So it's like number of aligned reads, proportion of reads that are aligned, number of duplicate reads, and so on. These are box plots of these QC measures stratified by batch. Okay, so each batch will have a box plot. And this is, for example, the percentage of mRNA bases, the median CV for the coverage. Okay, so what you see right away is the QC measures, they vary by batch. They also vary within batch. But more importantly is that these sample level QC measures are associated with the read counts or the expression measures. And this is shown in this scatter plot, which represents the first principal component of the QC measures versus the first principal component of the read counts. So you have substantial correlation. Same message in this plot, which look at the count principal component, first, second, third, all the way to fifth, and the absolute correlation of the count principal component with each of the QC measures. Okay, so substantial association of count and QC measures. So that's a little recap here. So sample level QC measures can vary substantially within and between batches or C1 runs. Some of these QC measures clearly point to low quality samples that should be filtered out. For example, if you have a tiny alignment rate. There can be a strong association between the QC measures and the read counts, from which we'll derive our gene expression measures. So it can be advisable in some cases to do a preliminary filtering of samples based on these QC measures. And it's also advisable to keep track of the QC measures and our normalization. Okay, now let's look at the gene level counts. These are box plots of the gene level log counts per cell. So again, 600 or so box plots, and they're colored by batch. Substantial variation in the log counts between cells and between batches. A better summary of the counts than the log counts is the count relative log expression. Okay, so the relative log expression of a gene in a particular cell is defined as the log ratio of the read count of that gene in the cell to the median count of that gene across all samples. If the log counts have similar distributions across cells, you would expect the RLEs to be centered at 0, and you would also expect to have the box plots that are lined up at the top and bottom, so similar spread for the log counts. So this is clearly not the case. Substantial variation in RLEs within and between batch. And also the strange shape here of the box plot is due to the excess of zeros. So we'll still need to deal with zeros in downstream analyses. Okay, so just a quick recap here before we jump to normalization. After gene and sample filtering and before normalization, large differences in gene level count distribution within and between batches, substantial zero inflation, association of counts and sample level QC measures. So normalization is essential before any downstream analyses like cluster analysis or differential expression analysis, because we want to make sure that when we see a difference in expression measure between samples or between cells, this difference is really due to differential expression and not a technical artifact. Okay, so the first main topic, normalization. And this is joint work with Nir Yosef's lab at Berkeley. So Davide Risso, my postdoc, and Michael Cole, PhD student in the Yosef lab. So we've developed this framework, scone, that stands for single cell overview of normalized expression, as a general framework for the normalization of single cell RNA-seq data. So scone examines a range of normalization methods, global scaling methods like DE-seq or TMM, full quantile normalization, RUV. So this is a normalization method that infers unknown unwanted factors of variation. It does factor analysis on a subset of control genes, essentially. It also implements methods that normalize for known factors of unwanted variation. So if you know that there are large C11 effect, you can normalize for these. And it returns performance metrics to evaluate the normalization methods, as well as a variety of numerical and graphical summaries of the normalized read counts and metrics. And we will be releasing the package scone through the Bioconductor project. In the meantime, you can download the package from Nir Yosef's GitHub repository at the following address. So this is the model that's underlying scone. It's a log linear model. Here you have your matrix of log counts, samples by genes, and it expresses the log counts in terms of 3 main quantities. The first quantity here would be the biological factors of interest. So if you're looking at 2 different cell types, you would have, as parameter of interest, a fold change between the cell types. And in orange and pink, we have the unwanted variation. And the unwanted variation was divided into 2 types: known factors of unwanted variation, like QC measures or batch effect, and unknown factors of unwanted variation. So this is what RUV deals with, and this is what a regression-based method on batch effect would deal with. So the goal of between sample normalization method is to estimate this unwanted part so that you can zoom in on the variation of interest, the biological variation. So we have a variety of normalization methods. We know that the method has an impact on the log count, but then are we improving things or not by normalizing, and which normalization method should we settle on? So with these goals in mind, we've developed a variety of metrics for ranking normalization procedures. The metrics fall into 3 main categories. The first 3 metrics here look at the clustering of the samples, either clustering by biological condition or by batch. Clustering by batch would be bad. Clustering by biological condition would be good. That's why it's green, green for good when large, and red, poor when large. We're also looking at correlation metrics, so correlation of the count principal component with QC measures or with known factors of variation of interest. And the third category of metrics looks at the distribution of the relative log expression. Okay, so we've applied scone to our P63 data set. We use it to apply and evaluate 172 normalization procedures. So among the normalization procedures that we consider are scaling methods. So no scaling at all, no normalization at all. DE-seq TMM. Full quantile normalization. It's not technically scaling, but we've included for software, for programming reason within the scaling methods. Normalizing for known unwanted factors of variation like batch effect and unknown factors via RUV, or also known factors via sample level QC measures, and adjusting or not for biology. And to select a normalization procedure, we use 2 types of ranking for the 172 methods. We rank them by the mean QC metric. So 8 metrics, so we look at the average metric for each of the 172 normalization method. And we also look at a weighted mean score, which is based on the eigenvalues of the QC metrics. And the best method picked by the unweighted score metric is full quantile normalization, followed by normalization based on the first 4 principal components of the QC measures, adjusting for biology. If we use the weighted mean score metric, we pick full quantile normalization followed by normalization for the first 2 principal components of the QC measures. So hopefully the following figures will give you a better sense of what scone does. So this is one of the main plots that returned by scone. It's a plot of the first 2 principal components of the performance scores. So we would start from a matrix that has 172 normalization methods, 8 metrics, and we do principal component analysis on that matrix. And this is the second PC versus the first PC. You have a dot per normalization method, so 172 points on that plot. And we've colored the points according to their mean score. Blue is a low mean score, bad. Yellow is a high mean score, good. And the biplot here, we represent the axes, so the metrics. Okay, so the good normalization methods according to scone are here, bad ones are here. And it seems that one of the metrics that drives the biplot is the correlation of the count principal components with factors of interest. So let's see how specific methods do in terms of the scores. So this is the same plot, second versus first principal component of the scores, but now highlighting 4 methods. So the method that was picked by scone according to the mean score in red, method that was picked by scone according to the weighted mean score in bluish color here, teal. In green, this is a method that we've used frequently in our data analysis. That's full quantile normalization followed by RUV right here. So all these 3 methods tend to do similarly in terms of the QC metrics. What if we had done no normalization at all? So this would be the purple dot here. So we would do poorly according to the performance metrics. So now we looked at the same kind of plot, but we colored the points by normalization method. Okay, so still 172 normalization method, but then we've highlighted in red the ones that there's no scaling, blue full quantile, green DE-seq, purple TMM. So the scaling method doesn't seem to impact much on the performance of the method. And that's not terribly surprising because due to the very large distribution of log counts within and between batch, we need to be more aggressive than simple scaling. However, if we look here at the same type of plot, but now highlighting the methods in terms of whether or not we adjust by biology or batch, we get a very different picture. In blue here, in the area where we have poor performance, we have the methods that adjust for batch without adjusting for biology. And they do very poorly. And one reason that is because we have this nesting of batch effects within biological effects. What that means is if we remove batch effects, we remove the biology as well. That's the problem with nesting. Okay? So this is something to avoid and to think, you have to think carefully about the design you have before you do any types of linear modeling or general linear modeling, because you can get rid completely of the biology in your normalization. The last 2 or 3 plots that I'll show for scone are here. So this is trying to relate the supervised normalization methods that normalize, for example, for QC measures to the unsupervised normalization methods like RUV. This is a scatter plot of the first principal component of the QC measures against RUV's factor of unwanted variation. Okay, supervised by QC, unsupervised. So there's a strong correlation between the 2 factors. So RUV tends to capture the variation that's due to QC. Okay, so let's look now at the distribution of the counts after normalization, and all the results that I will show in the remaining of the talk are based on the method that was picked by scone according to the weight and mean score. So that would be counts that were normalized by full quantile normalization, followed by normalization for the first 2 principal components of the QC measures. So these are box plots of the relative log expression for each cell, colored by batch. So we have a much better centering of the box plots at 0 and more similar spread in RLE. And this is the same display, but focusing only on the housekeeping genes. We had 700 or so housekeeping genes. So again, much better results than we had before normalization. And now let's see what happens in terms of the correlation of the normalized counts with the QC measures. So same type of plots as I've shown earlier. we have scatter plot of the first principal component of the QC measures versus the first principal component of the counts. Zero correlation. Okay, we've reduced by design, of course, because we normalize by QC. So the results are as intended. So just a few words to recap the situation with normalization. Unnormalized gene level counts exhibit large differences in distribution both within and between batch, and also association of counts and sample level QC measures. Different normalization approaches vary in performance according to scone and can lead to very different distributions of gene level counts and hence clustering and differential expression results. Global scaling methods are not aggressive enough. Biological effects tend to be dominated by nuisance technical effect, and then you have this problem with zeros. For example, the DEC scaling factors are computed only based on 5 genes out of 20,000 genes. Batch effect normalization can work in some settings. However, if you have nesting of batch effects within biological effect, you do very poorly because you wash away all the biological effects of interest if you don't adjust for biology. We found with this data set and with other data sets that full quantile normalization followed by QC-based normalization or RUV performs quite well. So the rest of the talk will be based on counts that were normalized by full quantile normalization and QC normalization. Okay, the second main topic of my presentation is clustering. And the main persons behind this project is my postdoc, David Eriso, and my colleague, Elizabeth Purnham. So our motivation for looking into the clustering issue once again is as follows. We wanted to have a clustering approach that was robust to the choice of samples or cells. Because we found that both hierarchical and partitioning methods tend to be sensitive to the choice of samples to be clustered. We found that in single-cell RNA-seq, there tend to be a lot of outlying samples or clusters, and these mask interesting substructure in the data, often requiring the successive pruning out of dominating cluster to get to the findings, to get to the finer structure. We wanted to have an algorithm that was robust to the choice of clustering algorithm and tuning parameters. We wanted to avoid focusing on the number of clusters K. The number of clusters K is a major tuning parameter of partitioning methods like partitioning aramidoids or PAM and K-means. Methods for selecting K like silhouette width tend to be sensitive to the choice of samples, normalization, and other tuning parameters. Particularly, they tend to be conservative. They give us low number of clusters, and they tend to capture only the coarse clustering structure. Also, the number of clusters K is often not of primary interest. So here, if we apply partitioning around medoids to our P633 data set, we retain only 2 clusters, and that's too coarse. That doesn't give us the resolution that we want. And another motivation for proposing a new clustering method was that we did not want to force samples into clusters. Some samples are outliers. They don't really belong to any cluster, and when you leave them in, you tend to have poor clustering results and poor interpretability of the clusters. Okay, so to address these issues, we turn to resampling-based sequential ensemble clustering methods. And our goal is to obtain stable and tight clusters. Ensemble clustering is nothing new, really. The concept has been used before in the past 15 or 20 years or so. And the main idea behind ensemble clustering is that you look at multiple clusterings obtained from different algorithms or the application of a given algorithm to resampled version of your data. And then you aggregate the different clusterings. Aggregating clustering is a way to improve stability. And you can think of this as the unsupervised analog of ensemble methods in supervised learning, like bagging, boosting, and random forests. So we implemented our method in an R package called Cluster Experiment and we've already submitted the package to Bioconductor projects. So you should be able to download it from the Devel version of Bioconductor. So here's the main ID. So RSeq stands for Resampling-Based Sequential Ensemble Clustering. So we start from a base clustering algorithm, like PAM or k-means, and associated tuning parameters, like the number of principal components, the number of clusters, or the distance matrix. And then we generate a single candidate clustering as follows. We use resampling-based clustering to find robust and tight clusters, and then sequential clustering to find stable clusters over a range of k. Okay, so that gives us one candidate. We repeat this to generate a collection of candidate clusterings using different base clustering algorithms and different tuning parameters. Okay, so you could do this, for example, with PAM and 50 principal component. Then you could repeat this with PAM and 25 principal component. You could do Euclidean distance, correlation distance, whatever choice you want. So we end up here with, say, 100 different clusterings of our cells. The next step is to generate a consensus clustering over these different candidates. The way we do this is by computing a co-clustering matrix. So it's a matrix that has samples by samples, and each entry would be the proportion of times the samples are clustered together. And then we apply another clustering step to this co-clustering matrix. Okay? And then the final step is to look at that consensus and say whether we still need to do some merging of clusters that are not different enough. Okay? Once you have your clusters, a natural question is to ask if we can find gene expression signatures for the clusters. So that's the differential expression step. Let me say a few words about the differential expression steps before I show you the results. Okay, so suppose we've applied our RSeq method to the P63 dataset and we end up with 10 different clusters. How can we find gene expression signatures between the clusters? If we have 2 clusters, it's kind of simple. You could do 2-sample t-test or a robust version of that. If we have 10 clusters, it's not quite obvious what the comparison should be. A first pass would be to look at F statistics. Okay? The problem with F statistics is that they tend to be sensitive to outlying sample, but more importantly, F statistics are not specific enough. They're not useful for interpreting differences between clusters because they look for any difference between any cluster. Okay? So instead of applying F statistic, we're going to turn to contrast, and that's a standard solution in generalized linear models. So we rely just on the standard GLM methodology that allows us to use all of the sample when testing specific contrasts. So the type of comparisons that we might do is to look for differences between pairs of clusters and then look at all possible pairs, or to look for differences between one cluster against all others, or to use the hierarchy to find which comparisons to make. Okay, so we can have a tree for the different clusters, and then we go up the tree and test for differences between sister nodes in the dendrogram. And to test for differences between sets of clusters, we use the empirical-based framework that's implemented in the package Lemma. And we use the VUM option to account for over-dispersion. That's a way to model the mean variance relationship of the log counts. So the methods, our clustering methods, are implemented in the cluster experiment package. There's a wrapper function called RSeq that just goes through all the different steps. But the steps are, again, to generate the collection of candidate clustering is the cluster many function. To aggregate the different clustering, we have the combine many function. And then to merge non-differential cluster, we have a make dendrogram and a merge cluster function. So we applied RSeq to the Olfactory Epithelium P63 dataset. We generated 22 candidate clustering by looking at 2 different types of dimensionally reduction. The first 25 of the first 50 principal components, we used Euclidean distance. We use PAM as the base cluster with 5 all the way to 15 clusters. And here are the results. So these is a way of representing or comparing many clusterings of the same samples. So the columns correspond to our 636 single cells, and the rows correspond to the 22 clusterings. So it's PAM with k equals 5 all the way to 15, and then 25 or 50 principal components. Okay, so you can see that there's some stability for some cluster, but still substantial variability here between clustering methods. So we have our 22 candidate clustering, so the next step is to aggregate them into a consensus. So to do this, we build a co-clustering matrix. So again, this would be a 636 by 636 matrix, whose entries correspond to the proportion of times 2 samples were clustered together. And that's what the matrix looks like. Okay, the diagonal is the identity, because a sample always clusters with itself. And we built a dendrogram based on this co-clustering matrix, and highlighted this returned to us 20 or so clusters, 29 clusters. Okay, they're ended in red. If you look here, it seems like we've clustered too much. Okay, so we're kind of in the setting, if you're familiar with CART, with classification and regression trees, remember how it works? It just generates a tree that has a lot of branches, and then it uses cross-validation to prune. So we're kind of borrowing these ideas here. We're generating many clustering, but then we go back and ask, are these clusters really different or not? Okay, so we're going to work up the tree and test for significant differences between sister nodes. So that's the merge step. And the merge steps basically here would use lemma and voom and test for differences at each of the nodes. And after doing the merge test, we retain only 8 clusters. We have the -1 cluster. These are the observations that we decide not to cluster because the co-clustering is not sufficient enough. Okay, so this is a plot of the count second versus first principal components, and we've colored the cells by the clustering results. So now, instead of having normalization methods in the plot, we have a dot per cell. So 636 cells, and the cluster assignment, 8 clusters. Next question is, can we find gene expression signatures for the clusters? So this is where we applied our contrast approach, the dendrogram contrast approach. So this is a heat map of the log counts for the genes, the top 269 genes that were found to be differentially expressed using the dendrogram contrast. So that's going up the tree and testing at each node, are you different or not? Okay, so you can see some clear patches here in the dendrogram showing that there are indeed suggestion of differential expression between the clusters and contrast-specific differences. If we had used the standard F statistic approach, we would not get a clear result. That's again because the F statistic is not specific. It would probably focus on one cluster that tends to be really different from the rest of the cluster. It would not allow us to get to the fine structure of the dendrogram. Okay, and then just, I think I have about 5 more minutes, right? Okay, very, very quickly through the last topic, which is the inference of cell lineages. So unfortunately, I'll have to skip most of the motivation, but this is in the setting where we want to find, identify the differentiation between cell lineages. Okay, so in many cases, cells undergo gradual transcriptional changes where the relationship between the states can be represented as a continuous lineage that's dependent upon an underlying spatial or temporal variable. And this representation has been referred to in the literature as pseudotemporal ordering. References in the topic are the monocle package of Trapnell et al., Embedder, Campbell et al., T-scan from G and G. So there's been already quite a few methods proposed for that setting. We had some trouble using these methods with our data set, so that's what motivated us to develop our own method called Slingshot. And how we proceed with Slingshot is to start from normalized gene expression measures and cell clusterings. And then our output would be cell lineages. These are subsets of ordered cell clusters, and cell pseudotypes. So for each lineage, we have an ordered sequence of cells and associated pseudotype. So the first step in our approach is dimensional reduction by principal component analysis. Then we build a minimum spanning tree on the cell clusters instead of on the cells themselves. So that gives us more robustness. We have a granularity parameter to identify outlying clusters. We can let the user specify the root and the leaf node or infer these automatically. Once we have the lineages, then we use principal curves to obtain the pseudotypes. And in the case of branching of lineages, we do a shrinkage step to obtain pseudotypes for the two different branches. So we applied, I'm sorry for going so quickly through the methodology. I just want to show you quickly some results. So we applied Slingshot to the P63 dataset. We looked at the first three principal components, and we were able to identify two lineages. The first lineage corresponded to the HBC to neurons transition, and the second to the HBC to sustentacular cells transition. Our next question was, can we identify genes that are differentially expressed in the two different lineages that characterize the transition from renewal to differentiation, and to differentiation for neurons or differentiation to sustentacular cells? So for differential expression analysis, we rely on a standard statistical framework of generalized additive models. We used GAMs to regress the log counts on the pseudotypes. And this analysis suggests that many genes are involved in the differentiation process. And we also got very different sets of genes for the two lineages, suggesting that you have distinct processes in neuronal versus non-neuronal lineages. So this is, actually I can probably jump to an interactive display. So this is when we fit the pseudotypes to the first three principal components of the counts, 1 point per cell here. And you have here your neuron cells. You start in the HBCs, and here the sustentacular cells are in the second branch. They're more closely together than the neuronal lineages. And the genes, when we apply a generalized additive model to find genes that are differentially expressed within a lineage, this is what we get for the first lineage. That's the HBC to neurons transition. These are the top 9 genes that were found to be differentially expressed. So regression of log count and pseudotypes. So you can clearly see some patterns here along that lineage. You have increasing of expression with pseudotype. This one you have, for this gene, you have decreasing expression with pseudotype. So I think I'll have to end here because of time. My apologies for rushing so quickly through the last part of the talk. But thank you very much for your attention. I'd be happy to take questions.
So we have time for